Yes, sir.
Right,
Yes, sir. Good afternoon, sir.
right. So yesterday we completed about the tissues, tissue types, meristematic tissue completed, and we have started yesterday permanent tissues, some parenchyma, collenchyma, now the sclerenchyma. So already you must have learned during practical training classes about the tissues, right? So anyway, let's discuss once again about the types of tissue, sclerenchyma. Sclerenchyma first discovered by Vittinus. It is a simple tissue. It is one of the simple tissue like parenchyma and collenchyma. Let's share. Right. Are you able to see the PPT slide? Hello? Slide visible or not?
Yes, sir. Visible.
Sclerenchyma types.
Yes, sir. Yes, sir.
Right. Before discussing about the types of sclerenchyma, let's see the some characteristic feature of sclerenchyma. It is sclerenchyma type of simple tissue, and it is when compared with rest of the simple tissue such as paren and collenchyma, this is seems to be very hard, and it has the lignified thicknings. If wall layers, the parenchyma mostly made up of abundantly consisting cellulose chemicals. In collenchyma cell wall, cellulose, hemicellulose, and pectin substances present. But if you see the sclerenchyma cell wall composition lignin, the lignin present in secondary walls. In sclerenchyma, just wait, let me take the marker. In sclerenchyma, for example, this is sclerenchyma cells, primary wall, both secondary and primary, both walls are present. Primary and secondary. But in parenchyma, collenchyma, only primary wall layer present. Only primary wall layer present. Actually, for example, this is primary and this is secondary. The secondary wall formation from the protoplast, which mean after formation secondary wall layer, there is no protoplast. That's why it is considered as a dead cell. That is the main difference between the parenchyma, collenchyma. Parenchyma, collenchyma, both are living cells because it consisting clear protoplasm. Whereas in sclerenchyma, since the composition of secondary cell wall with lignin, the completely protoplast disappear. That's why it is considered as a dead cells. And the function of sclerenchyma, like collenchyma, both are functioning as giving anchoring to the. Actually, these both are mechanical tissue. Gives the anchoring to the plant body. That's why the scleren and colon both functionally called as sterome. However, the sclerenchyma differs in general from the collenchyma presence of lignified secondary walls and absence of living protoplast. That is the difference between scleran and collenchyma. Right? If you see the types of the sclerenchyma, the mainly the broadly two categories: scleren, scleroids, and second one fibers, scleroids and fibers. First, the scleroids. Actually, the sclera when compared with fiber, the scleroids had the small in structure, and these are in hard, and which is found in the plant body. The embedded in the softer tissues, as seen in the flesh, mainly in the flesh fruit and bark of cinnamon, root, leaf mesophils. Such parts consisting scleroids. Based on the structure, these scleroids classified into brachyscleroids, macroscleroids, osteoscleroids, osteoscleroids and osteoscleroids and trichoscleroid. First, brachyscleroid, which is round in shape, macroscleroid, rod shaped, osteo, bone shaped, aster, star shaped, trichome shaped, trichoscleroid. Let's see the diagrams. Uh, right. Are you able to see the types of scleroids diagram? Hello?
Yes, sir.
Right. First, let me complete brachyscleroid. Just wait. Why are the lines? Right. First, brachyscleroid. So, just wait 1 minute. Brachyscleroids. Right. The brachyscleroids are shaped like a parenchyma cells. We can observe here the like parenchyma type cells, and the cells more or less isodiametric and polygonal in cross section. The brachyscleroids are commonly found in flesh edible parts of some fruits. The mainly fresh foods consisting this type of scleroids. And macroscleroids. Next one, macroscleroid. The macroscleroids are, we can observe in diagram. They are in elongated. They are look like a parenchyma cells like brachy. They are elongated and columnar structure. They are very thick walled cells and narrow lumen. Narrow lumen. That means if we see internally, the lumen appeared very narrow like this, duralumen, lumen, and elongated cells. Cells look like this. So this is macroscleroid. And this type of scleroids majorly found in epidermal cells of the seed coat, the mainly seed coats, and brachyscleroids mainly found in flesh, fruit, brachyscleroid, epidermis of seed coat, and next one, osteoscleroid. Osteoscleroid, we can see in diagram, osteoscleroid, as the name suggests, they are bone-shaped scleroid. Bone-shaped, they are elongated cells like a macro, but with bulged end like bones. They are like a macro, they also like a elongated, but the both poles, the bulges like a bone, like this. So this is called osteoscleroid. Osteosclerites are found in the seed coat of Pisum. So the osteosclerites, macroscleride, they both are found in the seed coat. Next, star-shaped scleride, hasteroscleride. They are star-shaped. The name suggests like that they have the lobes and arms radiating. We can observe the radiates like this, radiating. The number of arms radiates varies in different species. Hasterosclerides are frequently found in the petiole of nymphia. Next one, tricho, look like trichome, trichosclerides. They are air-like elongated sclerides. Sometimes these sclerides are branched. Mainly these are found in Holia and Musa. Musa leaf. Mainly these are found in the leaves. So this is all about types of sclerides. Sclerides, important for short answer. Is it clear? Any doubt in sclerides? Hello? Sclerides, okay? Josna, Swapna,
Yes,
Krishna
sir.
Priya, are you there?
Yes, sir.
Yes, sir.
Respond. When ask the question, please respond.
Okay, sir.
Right. So this is the simple sclerides. Now another type in sclerenchyma, broadly classified two types. The smaller one sclerides and somewhat larger one. These are considered as a, what is the next? Another part of sclerenchyma. Hello? What is the next part? Next, sclerides completed. Sclerenchyma,
Fiber.
broadly,
Fiber,
what?
sir.
Fibers. Right, next one, fibers. Uh, Actually, the fibers are second category of sclerenchyma cells in plants. Unlike scleroids, the fibers are much elongated. When compared with scleroids, they are much elongated cells with pointed ends. They are present in almost all parts of the all plants of the pods. Their cell lumen is very narrow. The fibers possess very thick and hard lignified secondary cell wall. They are the main mechanical support in plant. Apart from the collenchyma, sclerenchyma, the major mechanical support was given by fibers in plants. The fibers are associated with the vascular bundles as bundle cap or sheath. Fibers also forms elements of xylem, phloem as xylem fiber and phloem fiber. Now the classification of the fibers. Let's see classification of the fibers. Right. Classification of fiber. Before classification, let's see uh, the fiber structure. The fibers, as I said, are you able to see the classification of fiber slide? Hello?
Sir. Yes, sir. Sir.
Right, right. Are you follow marker?
Yes, sir.
Right. So this is like elongated structure, like actually, and more narrow than this diagram. So for our clear understanding, I had drawn like this. So this is lumen also very narrow. Primary wall, secondary wall, primary wall, secondary wall, and the center actually very narrow, very narrow. that is called lumen. No protoplast, no protoplast, only empty space is present internal, that is called as lumen. And this fibers passes very thick and hard lignified secondary wall. Secondary wall, as usual, consisting or composition with lignin. Lignin. So this is the structure of the fiber. Let's see the classification of fiber. The plant's fibers have been classified into two broad categories: xylem fiber and extraxillary fiber. Name itself given xylary fiber. Where are the xylary fiber? Where are the xylary fiber? The name itself given xylary, which means the fiber located in, hello, fibers located in
Xylem.
xylem.
Yes, sir.
Right, right. And they also called as wood fiber.
Wood
They also
fibers.
called as wood fibers, and different type of xylary fibers are in the plants. They are, just wait, they are trachid fibers and libriform fibers. And I will explain in detail about this both fibers. And but let me give the brief introduction about here. Right. Actually, the xylary fibers, one is fiber trachates, second one is libriform. I will export, remember these two words, fiber trachates and libriform. I will clearly explain later, but let me give the brief introduction about those two. They are first fiber trachates. Fiber trachates. The fiber trachates, actually these are transitional forms between the tracheids and the extreme fiber. And these fiber tracheids are longer than scleroids, but they are shorter than true xylem fiber. These are the intermediate, longer than scleroid, shorter than true fiber. They are the fiber tracheids. And another one is libriform fibers. Libriform fibers are highly specialized xylem fibers. They are long cell with narrow lumen like a true fiber. This libriform fiber possesses numerous slit-like pits on their secondary cell wall. Some pits also we can find on this libriform fiber. These two. And another one is uh, extraxillary fiber. Extraxillary fiber means, where are the extraxillary?
Phloem.
Not only phloem, and also present in fibers, cortex also.
Cortex also.
Right, right. So cortex and some are pericycle. So any other xylem region, maybe medullary fibers. They are the extraxillary fibers. First, they are the fabric located external to the xylem. Most commonly, these fibers found in phloem. First one, phloem fibers, and also found in cortex also. Let's discuss one by one. The first one, phloem fiber, and also called, these are called bast fibers. Phloem fibers, and another name, bast fibers. They are located in the primary and secondary phloem. I will explain later, primary and secondary. Primary phloem and secondary phloem of the vascular tissue. And next one, cortical fiber, cortex, which are present inside the cortex. They occur either single or groups. And the function, any fiber function, remember, provide the mechanical support in young plant pods. So these are the cortical fibers. And the fiber, any economic importance, the fiber economic importance in plant system, it is give the supporting. But any other uses in daily life?
Yes, sir. They are rich in roughages.
Rich in,
Actually, fibers are good for
right.
digestion.
But how it is useful in commercially?
Commercially, it means this
They
coconut
may be
fiber.
useful in making rope.
Useful.
Right, ropes.
Yes,
Making
sir.
ropes,
Coil.
and here the ropes also two types, hard and soft.
Yes,
Mechanically,
sir.
two types of fibers we get obtained. First, let's discuss hard fiber, which are mainly obtained from the monocot plants. They are very thick and lignified cell walls. They are hard and very stiff fiber. Example, coccus, coir. We can already, you must have seen this type of fiber, coir fiber, the mainly utilizing for rope making. And soft fiber, the soft fiber obtained from the dicot plant. Dicot plants, hard fiber from monocot plant. All soft fibers are bast fiber, phloem fibers. This is important. Why? Because why the hard fibers hard? These are hard, and why they are the soft? The first of all, the hard fibers mainly from the xylem fibers, from the xylem cells, and mostly the xylem cells are dead cells when compared with phloem. That's why it is very hard. But in phloem fibers, in phloem cells, they are in soft tissue, living cells. So these phloem fibers are very soft in nature and narrow, flexible, and also it gives shining. Example, jute fiber. Jute fiber, example for softness. So this is about sclerenchyma types, sclerenchyma tissue, and also sclerenchyma types. Is it clear? Any doubt in this simple question? Hello?
No, sir. Thank you.
Right. Now the another important one is now one end is complete. That is permanent. In permanent tissue, simple tissue complete, and meristematic tissue complete. Right? The first in tissue system, do you have table? Do you have table, tissue system table that I explained yesterday? Tell me the types of tissues. Types of tissues.
Meristematic
Yes.
tissue.
Right.
Systematic
Meristematic and permanent tissue. Right.
permanent
Yes,
issues.
sir.
Meristematic and permanent. Where it is? Just wait. Right. And again, permanent tissue. How many types?
3 types, sir.
No. Broadly
What
two
are
types are
those?
simple and complex, I think.
Collenchyma, parenchyma, collenchyma, sclerenchyma.
Those three comes under simple permanent
Parenchyma,
tissue.
collenchyma, sclerenchyma. And
Oh,
before that, parenchyma. Sir, before that actual, okay. The parenchyma, collenchyma, sclerenchyma belongs to which type of
Brown
tissue?
tissues. Brown tissues.
I
Sir,
think simple.
parenchyma, collenchyma, sclerenchyma comes under simple permanent tissue. I
Right.
think so.
Not permanent, simple tissue.
Simple
tissue.
tissue. Okay.
Simple. Yes, sir.
Simple tissue. What is the next
Yes, sir.
one?
Permanent.
What is what is the next in permanent tissue? In permanent tissue, simple tissue completed. In tissue, in orderly, meristematic tissue complete. And permanent tissue, actually permanent tissue broadly categorized into 3 types: simple tissue, complex tissue, and specialized tissues.
Yes, sir.
And in simple tissue, again classified into parenchyma, collenchyma, sclerenchyma. Right?
Sclerenchyma.
Now these 3 completed. parenchyma, collenchyma, sclerenchyma completed.
Yes, sir.
Now the complex tissue, right?
Yes, sir.
What is complex tissue?
Tissue which is made up of more than one type of cells.
Right, more than one type of cells, but structurally different, but not functional. Functional,
sir.
they are alike. What is the functional of, for example, xylem, one of the complex tissue, phloem, one of the complex tissue. Now tell
Yes,
me,
sir.
Yes.
what is the unique function of the xylem?
Unique function of xylem is to transport water and minerals.
Transport water
Transport
and
the
minerals.
water,
Transport water, minerals, and also gives the support to the plant body.
minerals.
Right. So this is the functions. Now let's. Are you able to see the for complex permanent tissue slide?
Yes.
Yes, sir.
Right. Is it clear?
Yes, sir.
Right. The complex tissue are made up of, as you said, more than. Please be in mute. If you have any doubt, ask me. But while I'm teaching, please be in mute. Thank you. So complex tissue, as you said, sorry. This tissue made up of more than one type of cells, but these work as a unit due to the composition of more than one. This is considered as heterogeneous, not homo, like simple tissue. The complex tissue, actually simple tissue, homogeneous, but the complex is heterogeneous because of different type of cells are there. The complex tissue are absent in gametophytes. Already already I explained the plant stages in during practical training classes. Gametophyte and sporophyte. What is the gametophyte? What is the sporophyte? Have explained already.
These are the two phases of the plant life cycle, sir.
Okay.
Those two can be acted as the alternation of generation
Okay.
in the plant life.
Where the gametophytic plant body dominated in plant kingdom?
Bryophytes.
Yes, exactly. And apart from bryophytes,
Rest of the plants can be dominated as sporophytes. Sporophyte plant body can exist in a higher plant.
no. Actual gametophyte dominated in, as you said, bryophyte.
Yes,
And
sir.
also another plant kingdom, algae also gametophyte.
Yes,
So
sir.
in
Yes,
this
sir.
both there is no complex tissue,
Yes,
right?
sir. Yes, sir.
Next, the complex permanent tissue, two types, xylem and phloem. The first, let's discuss about the xylem. Xylem word first discovered by Nageli. The function of xylem, already you know, the conduct of water and minerals to upward direction of the roots from the roots to the stem and leaves and give mechanical strength to the plant body. This is the unique function of the xylem, right? And complex permanent tissues on the basis of origin, the xylem is divided into two types. Xylem, primary xylem and secondary xylem. Primary means first initial stage. Secondary means second time. Let's see the primary xylem originated. These are originated from the procambium. On the basis of development, the primary xylem is divided into two types, protoxylem, metaxylem. Already you must have learnt during training classes, practical training classes during section cutting. What you observed in section cutting? Internal, what you observed in xylem? Where is procambium? Yesterday have explained where is procambium. So primary xylem, so what is given here? In origin of xylem, primary xylem clearly given primary and secondary. The primary xylem originated from the procambium cells. What is procambium? Where it is? Hello. Yesterday itself we completed about the procambium. Please actively interact in this class.
Sir, I was absent, sir, yesterday, so that I am unable to.
What?
I was absent, sir, yesterday, so that
Even
I.
I am absent, sir,
Of course you
yesterday.
are absent, but you must have seen in online, that means in YouTube. You should have observed.
I think it is present between xylem and phloem, sir.
That's it. That's it. So, right. If you take
Thank
the
you.
vascular bundle, are you able to see the marker diagram? Hello?
Yes, sir.
Are you follow the marker?
Yes, sir. It is visible.
Right. So, let me change the color. The blue color is
Procambium.
procambium.
Yes, sir.
This is actually initial stages, formed in initial stage before secondary growth. Right? This is
Yes, sir.
procambium. From the procambium, the primary xylem development here the given, right? So in primary xylem also two parts, protoxylem and metaxylem. What is protoxylem? The cells of protoxylems are small as compared to metaxylem. That means actually this is the procambium cells. What happened in procambium cells?
Either side they will divide it and then.
Right. I will separate one of the procambium cell here. Right. So one division, one plane division like this formation. Two cells are formed like this, and this is one developed into one cell. Again divided into another plane, another cells. Again divided, again divided. So the always formation like this. After division takes place, after division group of cells, but development variously developed. For example, this is the first develop. This is first develop. Later this one, and finally this one. That's why the final development in small in structure. The first developed is large in structure. So this is the first protoxylem, first formed. This one small structure called protoxylem. Large one called as metaxylem. So this both protoxylem small and metaxylem large, the both are development from procambium cells. Procambium cells. So anyway, this total complete called as primary xylem. This total completely called as primary xylem. So in primary xylem, first formed xylem is protoxylem and final xylem is metaxylem. When compared with proto, metaxylem large in size. Is it clear? Hello?
Yes, sir.
Yes, sir.
Right. Now, next one. Right. Secondary xylem originated from the vascular cambium. This is very, very important. Vascular, where is vascular cambium? Actually, this is vascular cambium. You know procambium, right? So secondary xylem formed from the vascular cambium. Where is vascular cambium? Actually, this vascular cambium developed before secondary growth. Before secondary growth, I will explain clearly in secondary growth. But remember the word vascular cambium. Actual cambium ring. After formation cambium ring, from the cambium ring, secondary xylem cells are originated. Actual primary xylem actually development from the procambium cells, which is procambium is the part of meristematic tissue, but not but vascular cambium not from the meristematic tissue. From permanent tissue, the cambium ring formed, and they are developed into they are forms as a secondary xylem. Secondary xylem. I will clearly explain during when I explain the secondary growth. Okay, but remember the word protoxylem from procambium. Sorry, primary xylem from procambium, secondary xylem from vascular cambium. Okay, hello, is it clear?
Yes, sir.
Right. And now let's see the elements of xylem. Majorly four elements: xylem tracheids, xylem vessels, xylem fiber, and xylem parenchyma. In these three, so in these four, three are
Dead tissue.
tracheids, vessels, fibers, or dead tissue. Only xylem parenchyma is living as living. Now let's discuss one by one. And before that, some brief introduction. Tracheids majorly found in the primitive plants, primitive vascular plants. Xylem vessels found in higher vascular plants. And let's discuss one by one. Xylem tracheids. So xylem elements. Are you able to see the diagram?
Yes, sir.
So this is the microscopic view, electron microscope view of the vessels and tracheids. These are the vessels, long tracheids. These are the tracheids, elongated, look like a fiber, like structures, tracheids, and narrow, elongated, and vessel edgement somewhat broad when compared with tracheids. You can observe in diagrammatically clear. Can you compare the both? Hello, both diagram. Can you compare the both diagrammatically? Xylem. Hello,
Yes, sir.
right. So when compared with tracheids and xylem vessels, tracheids are elongated and slender. Elongated look like a fibers, linear shape, and the basic difference between these two types cells is that tracheids are long, linear, angular. Their end walls, actually end walls, tapered end walls. But vessels, when compared with tracheids, short in size, wide and drum-like structure. End walls opening with termed perforations, but not blunted end. Sharp blunted end present in tracheids, but perforation present in vessel. The tracheids is the fundamental element of the xylem tissue and considered more primitive. Already told you more primitive than the vessel, and the vessels are the advanced character. So the term vessel refers to collectively the series of interconnected cells joined to end like a ladder, one cell above another. The xylem vessel also called as trachea. The another name of xylem vessel called as trachea. And in xylem vessels, we found the perforation plates. We will see through the perforation plates, the water move. And here, these are perforation plates here. This is perforation plate. So actually, this is one perforation plate. Next, another vessel cell. So water move from through this direction. If we observe in tracheids how the water move in tracheids, the water move through the pits in cross, like a cross section like this. Actually here, Alder told you the tracheids are with tapering ends. Alder told you like this with tapering ends, and this is the sharp tapering ends. One tracheid, and let's change color. Another tracheid, the green color. Arrange to this another tracheids like this. So where the pits are present, where the pits instead of perforation plates, pits are present. Through the pits, through these pits presented cross walls and the cell wall layers. Through the pits, water move from one cell to the one tracheid cell to the another tracheid cell. Right here, the water move from the perforation plates to upright direction. Right? Is it clear?
Sir, I have a doubt.
Hmm.
In this trachid, sir, water can it come out of the trachid cells, sir? Trachid cells are there, no? They will be like
No,
cross. Sir,
of course. Some
pits are there.
lateral movement also occurs sometimes.
That
The
is true.
lateral movements of occur, but what are present in lateral side? The lateral side present maybe xylem vessels or xylem parenchyma cells. They cannot leak out from the plant body.
No,
Hello?
sir, my point is, vessel elements can exchange the water, no, sir, in between them.
Vessel elements.
Vessel elements, suppose if two are there,
Okay.
in between them, water can be interchanged, no, sir.
Yes, exactly.
Is it possible in tracheids to come out water out of the tracheids cells, sir? Like through pits. Pits are highlighted, no, there, sir. Pits are there, no?
Yes, pits are there. Okay.
From that pits, can water come out of the cells, or else just they can interchange between two cells only?
Interchange between two cells, between two tracheids. Actually, these
That
pits connecting to the adjacent tracheid cells.
okay.
These pits actually the pits connected to the adjacent tracheid cells.
Okay, sir. Okay, sir. Thank you.
Right. And now next one. Uh, now your doubt is clear. It is element xylem, components of xylem here. Are you able to see this diagram, pits diagram, vessel diagram? Hello?
Yes, sir. It is visible. Yes, sir.
Right. So actually, so these are the perforation plates. Where is pen? This is perforation plates. These are the pits. Actually, pits present at the wall. entire wall pits are present, whether it is simple pits or border pits. And also one vessel to another vessel, there is a connection, that is the perforation connection. Perforation plate is present. This is one vessel, and above another vessel, and above another vessel, like this. And the same tracheids, the same pits are present, but there is no perforation plate. And the arrangement of these tracheids like a parallel, but not one above the another, like parallel. Actually, this arrangement is closely arranged, and also these pits also interconnected, but it is not a 3D diagram. It's not a 3D diagram. Just assume these two pits are interconnected. Pits are interconnected. So the water moves through this pits layer, one trachyte to the another trachyte layer. This is the connection. The pits are the connection between the tracheids. Okay. Next one.
Yes.
Uh, the elements of xylem, tracheids and vessels completed. And in this xylem vessel pits are there. These are simple pits and border pits. And let me explain about the simple pit. If you see the simple pit, this is just a structural diagram. If you see in detail in detail the simple pits, let me take marker, right? Middle lamella, first one, either side, primary wall layer. Let me change color. Red, primary wall layer, and secondary wall layer either side. Another color blue. This is the wall layer, and somewhere they leave the gaps. Like this. Only middle lamella present. Like here. So this is in cross view. This is appear as a round, like this, like structure. Round. So this is the simple pore. And next one, border pits. This is the border pits diagram. If you see the border pits, the same middle lamella, and in between the bulges like part is present, that is called torus. Torus. Middle lamella. Apart from the middle lamella, primary cell wall also there. Primary cell wall, but secondary cell wall not uniform. The secondary cell wall look like a harrow-like, bow-like shape, like this. Actually, middle lamella, either side, primary, but in second lamella, second wall layers, bulges like structure, like not a uniform, like simple pore. The bulges like structure formed. This bulges like structure, the in between present porous present. This pit called as border pits. That means simple. Just imagine the secondary wall layer like this. Some bulges like structure. Here also same. Secondary wall, only secondary wall layer, bulges like a arrow, sorry, bow, like a bow shape, right? So this is called border pits. And perforation, the where it is secondary wall layer, spitting, right? Other category, right, right. So in border pits, two types, in pitting, two types of pits, simple pits or border pits. Right. So next one. So what is the difference between simple and border pits? Where the difference? What you find out difference between simple and border? Hello?
Yeah, simple pits are not surrounded by secondary cell wall,
Right.
whereas border pits are the pit is surrounded by secondary cell wall, sir.
Right. Secondary cell wall somewhat bulges like and bow shaped, and it is columns like torus present in middle layer. That is the main difference between simple and border pits. And next one.
Sir, I have a doubt.
Oh,
Is that purpose? What is the purpose of this, sir? To exchange the things, sir, between two cells.
simple and border pits.
Function.
What is the function of pits first of all?
I think they exchange the materials between two cells.
Which material?
Uh, okay. Water.
Yes, that's it.
Okay.
Right. Next one. So now let's discuss one by one again the xylem tracheids. Already these points all discussed. Xylem tracheids is a primitive conducting tissue. They are unicellular having narrow lumen but wider than xylem fiber. One difference between xylem fiber. Let me take marker. When I give introduction about xylem tracheids, I said xylem tracheids look like a fibers, right? But what is the difference between fibers and the tracheids? It is the simple difference of both are unicell but having narrow lumen, but wider than xylem fiber. This lumen in tracheids wider, but in fiber very narrow. And the tracheids join together from their ends to form the longer rows. These rows extended from the roots via the stem to the leaf. The transverse septum lies between the two tracheids. Already told you the connection with help of the pits, and with help of the tracheids, water move from the one trachea to another trachea through their pits. The pits are uniligniformed areas on lignified walls. Due to the transverse septum, lumen is discontinuation to tracheids, but in vessels continuation. But what happened in tracheids? Always the cross septa formed to one tracheids to another trachea. They are dead and lignified common, deposition lignin common, and also have the different type of thickening also given in our material. Different type of thickening, scalariform, ladder, simple, whatever it may be given, the different type of thickenings are there. Usually border pits are present end wall of the tracheids. Already we discussed difference between border and simple pit. Maximum border pits are found in the tracheids of gymnosperm plant. When compared with angiosperms, gymnosperm border pits are high. Maximum deposition lignin found is pitted type of thickening. Annular, spiral, they are different type of thickenings. No need explain about that. Reticulate, already clearly given, diagrammatically given in our book. Diagrammatically given. And also see here, types of thickening here. First annular, lines, spiral, S shape. Can I draw clearly or is it clear? Hello? This trachea techniques clear, diagrammatically clear. Or should I draw? Concept clear or not?
Yeah, it is okay, sir. We are observing in the textbook
Right.
also.
Simple, annular, just lines, spiral, like a shape. Let's give the rough. The simple say sorry. S-shape thickenings, like thickenings are S-shape. Ladder, scalariform like a ladder, and pitted thickenings. Pits, some now and there pits are there. So this is about different type of thickening present in the tracheids. And function of tracheids already you must have knew about this. Xylem vessels. When compared with the tracheids advanced one, the basic structure of vessels same as tracheid. The vessels multicellular, but tracheids uni. Why it is uni tracheids? Why the uni? Because the tracheids have the cross septa, which separate the one tracheid to another. That's why it is discontinued. Tracheids are discontinued, but not vessels. Vessels continued like multicell. The lumen of vessels wider than tracheid, and the vessels of lumen wider than fiber or not. Hello. The lumen, for example, here I would like to ask one simple question to you. The lumen present in three types of cells: fibers lumen present, tracheid lumen present, and vessels lumen present. What is the difference in these three lumen?
Vessels lumen is broader than other than two, sir.
Right. First border, larger one is the vessels.
Vessels.
Next it follows tracheid.
Tracheids. Last one
Right.
is fiber. Yes.
Right. So, and also when you draw these three diagrams, you should have show that type of difference also. Okay.
Okay, sir.
Right. And transfer septa absent. Here clearly transfer septa absent between two vessel elements, and if the present, then it will be porous. That means perforated plates. Already the end walls perforated. Actual septa, but the septa with perforated. As the perforated, it is a continuation. And vessels are more capable for conduction of water than tracheids. Why it is more capable? Why the vessels more capable than tracheids?
They
Can you
have
guess?
more large. They have larger lumen.
Larger lumen, okay, that's it. And also it's continuation. There is no septa.
Yes.
There is
Yes,
no
sir.
cross
Yes,
septa.
sir. There is no septum.
Right. And xylem vessels, due to the presence of perforated end walls, vessels work as a pipeline. During conducting water, there is no interruption. There is no interruption. Vessel contain usually simple pits at their lateral wall. What about the tracheids? Which type of pits are there in tracheids? Generally, how many types of pits? How many types of pits?
Simple and bordered.
Right, just 1 minute. Right. Simple and border. And in vessels mainly, which type of pits are found?
Simple pits.
Simple pits, mainly simple pits found in vessels, but in tracheids, border pits. The
Okay.
thickening type of the walls is the same as tracheids. So vessels are only found in xylem of angiosperm. only xylem of angiosperm, but exceptionality is also present in some gymnosperms. That is when higher, well-developed gymnosperms such as ephedra, netum, welwitschia. Of course, these are the gymnosperms, but this consisting the vessels. What about pteridophyta and pteridophyta vessels present or absent?
Absent, sir.
Absent, simple. Pteridophyta, the more primitive than gymnosperms. That's why
Yes, sir.
only tracheids are present. That's it, simple. So the vessels are absent in some angiosperms. This is important. Actually, the previously just I said vessels mainly present in angiosperms. Of course, in some angiosperms, vessels less, such as Dracena, Yucca, and Dremis. Dracena, Yucca, Dremis. These are all angiosperms, but there is no vessels. And there are some angiosperm families in which vessels less angiosperms included Winteraceae family. Vessels as dead cells, syncyte. The cells which is formed by fusion of cells is called syncyte. So vessels as a dead syncyte, that is coalition, fusion, one cell to another. Fusion of cells, one vessel cell, another vessel cell, fusion combinedly form the syncyte. Tracheids and vessels are collectively known as water conducting elements or hard drum. Another name, both hard drum. That means hard drum means tracheids and vessels. The term hard drum proposed by Haberland. The types of vessel thickening, the same also here. The annular, spiral, scariform, petricular, same like a tracheid thickening. Same like a tracheid thickenings. Types of vessel thickening again given here, repeated. Function of vessel same. The same as. And also the examination point of view, difference also important. Tracheids vessel different. Short answer, very, very important. Should I explain? Already we discussed. I think no need. Should I explain tracheids and vessel difference? Hello? You tell me what are the difference between tracheid and vessels? These two completed. Now tell me. In first structural view. Hello? Voice clear or not? Hello? Are you there?
Yes, sir.
Right. You tell me the tracheids and vessel differences. Now tell me. Tracheid vessel differences. Tracheid vessel different, important in exam point of view. You learned separately about the tracheids, separately about the vessels. What are the structural changes in both? Tell me.
So vessels lumen is broader than the bracket, sir.
Okay, not only lumen, actually in terms of
Shape
cell.
also. I am coming to there,
Yes.
sir. Next
Yes.
one is
Yes.
vessels are connected to each other,
Yes.
whereas tracheids are not connected. Vessels can conduct more water than the tracheids.
All right.
Yes, sir.
Absolutely. And in terms of pits, what type of pits present in both?
Simple pits, sir.
Simple pits in which type of
Simple
vessel?
pits are present in vessels, sir.
Right.
Border pits are present in tracheids, sir.
Tracheids majorly found in which plant?
Pteridophytes, sir.
Pteridot and
Primitive
gymno.
plants.
Right. Pteridot and gymno. And vessels
Pteridot and gymno.
majorly found in angio.
Angiosperms, except Dracena.
Right. Already xylem fibers, about xylem fibers completed.
Yes, sir.
Right. Hello. Please be silent. Thank you. Now the xylem fibers already completed. They provide the strength to trachea and vessels, mainly these fibers. Fiber tracheids and libriform fibers also completed. And just brief difference between fiber tracheate and libriform fibers. Fiber tracheids, actually these are not a true fiber, not a true tracheate. Already the beginning I told you, these are the intermediate type fibers, intermediate from the fibers and tracheids. But libriform true fibers develop from the tracheids by increasing the thickness of the wall, of the decreasing the lumen diameter and cell length. And it provides fibers, libriform fibers provide the uh, mechanical support. Of course, this fiber tracheids also provide mechanical support. Now, xylem parenchyma, another element, xylem parenchyma. In terms of structurally, the cell wall is made up of cellulose. What about vessel and tracheid? The what type of chemical present in cell wall layer?
Lignin.
Right. But in parenchyma cellulose present, and it stores parenchyma function. Parenchyma function, the storage stores starches, starch and fat and tannins. And also it useful in radial conduction, lateral conduction of water. That is the function of xylem parenchyma cells. And xylem parenchyma not only storage, and also it is involved for lateral conduction of water, lateral movement. And this xylem parenchyma wall layer also possessing pits. The present in primary as well as secondary xylem. Already told you the xylems, primary xylem and secondary xylem. These parenchyma type of cells present in primary and as well as secondary xylem. And the cells were thin, but in tracheid vessels are thick, non-lignified in primary xylem, but slightly lignified in the secondary xylem. If when it is reached maturity, the some very less amount of secondary wall formed with lignified chemicals, and medullary rays or radial parenchyma also present, which help in radial conduction. The parenchyma present in secondary xylem is two types. The parenchyma present, that means xylem parenchyma, primary xylem as well secondary xylem. But in secondary xylem, two type of parenchyma there. One is wood parenchyma, ray parenchyma. Wood parenchyma, fusiform cambial initials. Ray parenchyma, ray initial of cambiums. Both cells are living, have thin walls. And here given the protoxylem, metaxylem, how the protoxylem formed, the metaxylem formed, already told you. From the procambium cells, primary xylem formed. From inside the primary xylem, two type of xylem, protoxylem or metaxylem. Protoxylem small size and first formed primary xylem. Metaxylem large formed and later formed primary xylem. And second one, it appears first and mature before. It appears first and matures before the organ completes its elongation. Appears only after the protoxylem has completed its differentiation, right? Tracheids and vessels commonly have the annular spiral thickening. Tracheids and vessels have the scleriform thickening. It is not important one. Trachea is by Aristotle. As more parenchyma, less trachea. Tylosis present, tylosis absent. More parenchyma trachea elements usually narrow. Tylosis, right. Xylem fiber part, xylem parenchyma. This is metaxylem, large. Metaxylem vessel. large. Already told you, larger size. Protoxylem, small. TSF xylem. Now the endark, exarch, mesorch. This is important in internal structure. Endark, exarch, mesorch. Is there any diagram? Right, not given. Let me take. For example, this is vascular bundle. This center region. This is center. For example, this is the steel center. Endark means first form protoxylem inside. This is endark condition. This development called centrifugal development. Development toward the center to peripherally. Eggs are present in centripetal. Centripetal means if the center C for center. Reverse. Eggs are outside. Let's change color. First form outside. Development toward the center. Right. So this is exarch condition. This is development like this. When mesorch, you can assume mesorch. The both type of development. That means protoxylem, here protoxylem, either side metaxylem. Here metaxylem, here also same metaxylem. Now this is mesorch condition present in uh, ferns and also leaf. Function of xylem. Already you know the xylem functions, no need to discuss about that. Right, clear about the xylem element? Hello? Hello?
Yes, sir.
Right. Now the phloem elements. Are you able to see the PPT slide, phloem?
Yes,
Hello?
sir. Yes,
So the
sir.
phloem, right. Phloem, leptome, another name leptome. Like the xylem also, like the xylem element, the phloem also have the different type of element, such as sieve elements, such as sieve cell, sieve tube, sieve tube, and also sieve cells also there, but not in diagram. But remember sieve cells, sieve tube, phloem parenchyma, companion cells, and phloem fiber. And phloem fiber also present. So in this only the phloem fiber dead cell. Except the phloem fiber, the rest of the all are living cells. Let's discuss one by one. First, sieve cells. Just compare with tracheids. Are you follow me?
Yes, sir.
Right. So the sieve cells, like a tracheids, they are single, narrow, and somewhat elongated cell with end walls pointed. Is there any difference between tracheids and the sieve cells here in these characters? Hello? Just compare with tracheid. Xylem cells are tracheid. Diagrammatically, the both are single. Both are narrow, elongated cells, end wall pointed. The sieve cells joined end to end, wherein they're inclined. That means the cross end walls overlapped. They possess special sieve-like areas with pores in the lateral and sometimes in the terminal walls called the sieve areas. Sieve areas are present. Sieve areas. The type of areas absent in xylem tracheids. Just compare with tracheids. Through this plasmodiasmata interconnected through protoplast to adjacent cell wall through the sieve area. The sieve areas are comparable to the primary pit field with the plasmodiasmata. The simple somewhat narrow like a, and here somewhere areas are there and another with sieve cells like this. This is sieve area. Through the sieve area, plasmodesmata connected to one cell to another, right? And this is one separate cell here. From this one separate sieve cell and another above another separate sieve cells. Is it clear? Hello?
Yes, sir.
Right. I will clearly show the sieve area diagrammatically. Right. Next, sieve tubes. The sieve tubes, the similar to the in xylem, which cells? Tube name
Fibers.
given. Right. No. Tubes. Where the tube-like structure in xylem?
Vessels, sir.
Right. That's
Vessels,
it.
sir.
Right. The same character. Say here the tubes are composed longitudinal files of cells. Each of these cells, the sieve tubes, usually they are near the end of the wall, highly specialized sieve areas. Sieve areas with relatively larger pores present. These are called sieve plates. Instead of sieve areas, the with pores sieve plates are there, the end walls. And also when compared with the sieve cells, these are somewhat drum-shaped structure, elongated. Sieve cells and sieve tubes differ in that form show only sieve areas, whereas the latter possess sieve plates. Only difference in end walls, one is sieve areas, another one is sieve plate. The sieve tubes of the phloem are comparable to the vessel of the xylem. I already told you. So this is the sieve plate here. This is sieve plate. So for the difference is here the from sieve plates, Carlos thickening. Here Carlos thickening in between. I will show the diagrammatically later. Carlos thickening present. I will show later. So this is look like a vessel, border shape. I will explain later about the sieve tube, sieve plate and sieve areas diagrammatically later. And next one, phloem parenchyma. This is parenchyma cells look like similar to the xylem parenchyma. The function also same. And if you see, if we observe the nucleus in initial stages in parenchyma cells, and also on the either side companion cells also there. Companion cells. What is the function of companion here? Not given here companion cells, but the companion cells also present in phloem, and these are made up of parenchyma cells and broad enlarged structure, and this companion cells function. 1 minute. The storage, it is act look like apart from the parenchyma, it is also involved in storage of food material and also lateral movement of the transport of the food material. And the component cell transport of substances in the phloem requires energy. One or more component cells attached to each other, each sieve tube provide this energy. It's been indirectly involved in transport. And not given in our book about component cells. Right. The
Sir,
components, right, tell me.
in this picture, where is sieve cells, sir? Just now you have highlighted a sieve cells.
Actually, sieve cells not given in this diagram.
Yes,
That's
sir.
why I have drawn the sieve cells.
Okay,
Simple.
separately drawn.
See, yes, already drawn. Should
Yes,
I
sir.
draw again?
No need, sir. I have seen.
Just compare with trachea cells. The only
Yes, sir.
different is sieve area present at the end of the wall. That is the difference with tracheids. The companion cells are tell me.
Okay, sir. Thank you. Thank you, sir. Thank you.
Right. Right. And next P type of cells are phloem protein, flea proteins. And next one. Where it is. So this is sieve plate in sieve vessels, sieve tubes. This is the pore. This pore either side thickening with collows thickening. Here collows thickening. This is if there is no collows, that is the sieve area. For example, this is empty sieve area. The plate formation with help of collows. This is collows. This is collows. And this is collows thickening. Now this is called as a plate. Now plasmodesmata through this plate. Through plasmodesmata, the food material substances movement to one cell to the another cell. And the proteinaceous substances characteristically present in the sieve tube element. That is called P protein. The protein appears in the form of compact structure, formerly called as slime bodies. They appear in the several morphological forms. The any type of cells like tubular, fibrillar, granular. The P protein is common in dicotyledons but uncommon in monocotyledons and gymnosperms. It spreads cytoplasm, continuity, disintegration, the nucleus. And the P protein, it consists of collows. The collows is characteristic component of the sieve elements. It is associated with the sieve plate, sieve areas in sieve elements just now seen. The collows is an unbranched chemical. It is unbranched beta 1,3 linked glucan. It is not uh, when it is staining, stained readily with aniline blue, reorcinine blue. To identify the colors, we stain with aniline blue or resorcinine blue. It forms the sheath around the connecting strands. You can observe in diagram that pass through the sieve pores and sieve pores of sieve plate. Already given diagrammatical difference of the sieve areas and sieve plates, sieve pores. Colors accumulated rapidly in response to injures. The killing the plant material for microscopic study involved the honing. So that means what happened actually if any injuries occur in phloem vessels, in phloem elements, if any injuries occur, automatically the P protein, proteinous substance of P protein activated it which produce the mucilage structure. This mucilage structure prevent the damaging of the existing and neighboring cells of the phloem elements. So that is the function of P protein. Another function of P protein, the prevent the damage of the cells in phloem elements. So this is about colors and the P protein. The last one, special tissue. I will explain later about the special tissue. So far clear? Hello? Hello?
Sir.
Today class clear or any doubt? Clear and kaima. Complex tissue, both xylem and phloem.
Yes, sir. It is clear, sir.
The phloem class somewhat fast when compared with xylem because the all characters similar to xylem. That's why I had to the wind up early. Is it okay?
Yes, sir. It is okay. Sir, can you send this PDF in the group, sir?